Welcome to this presentation entitled Recording Clinical Phenotypes Using SNOMED CT and FHIR. I'm Ian Green from SNOMED International. I'm the Customer Relations Lead for Europe and the Clinical Engagement Manager. And my colleague is Alejandro Metke from Cesaro in Australia.
Thanks, Ian.
So we'll start off really just with a couple of slides to reinforce some points about SNOMED. I'm sure you all know this, but what is SNOMED? So SNOMED is the world's most comprehensive multilingual clinical language. As a resource, it's comprehensive, it's scientifically validated, and provides clinical content. It enables consistent representation of clinical content from processing in electronic health records, and most importantly, in this case, it provides maps to other international standards such as ICD-10, GMDN, ICPC-2 to mention but some. It's used currently in more than 80 countries globally. There are three main components to SNOMED, which I'm sure you're all aware of. Those are concepts, descriptions, and relationships. The descriptions obviously give the human readable terms. The relationships provide the modeling. So when you talk about SNOMED in clinical use, you can view it in various levels of abstraction. The very detailed use, which is that concepts on the right-hand side of the screen, give you really detailed descriptions of clinical concepts and are used by clinicians at the point of care where they require that level of detail. But also on the left-hand side, SNOMED also provides some higher level grouping terms, which are very useful, both for terminologists and data analysis. And then in the middle, there's some middle terms which connect both the left and the right. So why is SNOMED CT important for genomic researchers? Well, patient health records can provide a wealth of information useful to genomic researchers around symptoms, around clinical findings, social factors, and other issues. The data needs to be in a format that supports the data abstraction and analysis. So SNOMED provides broad coverage across all healthcare domains. It provides a structured terminology, it has an ontological basis. It supports synonymy, which is important. It supports multiple languages, and therefore supports the global exchange of data. So the vision of the future as we move from genomics medicine into a new world of precision medicine, we're looking at the electronic health record, the EHR, containing a lot of detail, particularly around clinical phenotypes, which are required by genomics researchers. SNOMED will allow them to abstract that level of detail from the EHR and do genomics research based on that information. By them doing that research, they can then develop treatment algorithms which are specific to individuals or groups. And these can then be fed back into the EHR using the structures of SNOMED. This will then provide personalized or precision medicine, which benefits the patient in the longer term. So proving SNOMED's ability to support genomics, SNOMED International itself is committed to developing SNOMED CT to support the genomics use case. And to do this, we've partnered with Cesaro in Australia to undertake some initial pilots to explore the utility of SNOMED CT in the genomics space. We aim to identify usability, but also gaps within SNOMED CT coverage. And we ran two pilots. So pilot one is around genetic kidney disease, which was a smaller scale pilot. And then pilot two is cardiovascular disease, which is a far larger population-based pilot. And at this point, I'm going to hand over to Alejandro, my colleague.
Thank you, Ian. So before I talk about the pilots, I'm going to give a little bit of background about Australian Genomics, which is the organization where we conducted the pilots, and I will also talk a little bit about the technology that was used in the pilots. So as you can see from this slide, Australian Genomics is organized kind of in two axes. So in the first axis, you have the programs of work. These programs tackle different issues that are relevant in genomics. So for example, you'll see in program 1, we try to deal with the diagnostic and research and improving those in certain genomic areas. I am part, so CSRO is part of program 2. And our main goal in this program is to have define a national approach to do data federation and analysis. So this pretty much means linking genomic data with the corresponding clinical data. We also have additional programs that deal with very important aspects of genomics, such as health economics and also education and bringing the workforce up to speed. That's one of the axes, and as you can see, also the other axis is where actually the clinical research is being done. So the entities doing this clinical work are called the flagships, and we have mostly rare disease flagships and cancer flagships, which are kind of the areas where genomics is most useful at this stage. So as I was mentioning before, the flagships actually cover within these two main categories, cover 18 different clinical areas, and we have over 50 hospitals involved in these projects. These are all have human ethics committees, approving ethics for the entire program, and the data is collected centrally to support the implementation and the evaluation. As Ian mentioned, we have 2 pilots with SNOMED International. The first one is with the cardiovascular flagship, and the second one is with the renal genetics flagship. So, the technology that we used in the pilots, I'm going to briefly describe some of the main components, and then I will go into a bit more detail around each one of them. So the first piece of technology we used that it's, I guess, central to the work we did with SNOMED is OntoServer. So OntoServer is the CSIRO terminology server and implements the FHIR terminology standard. So of course, OntoServer has first-class support for SNOMED CT and also supports additional ontologies that might be used in the area of genomics. The second technology that's relevant for these pilots is REDCap. And for those of you who are not familiar with REDCap, it is an electronic data management system that is very flexible. So it allows creating forms very easily. And it is used by all the clinical flagships in Australian Genomics to enroll patients, to capture clinical and trial data, and to support evaluation. One of the key bits of technology that we developed as part of the work with Australian Genomics is called Redmatch. It was formerly known as FHIRcap, but we actually couldn't use FHIR as part of the name, so we renamed it. And this is an open source tool that allows extracting data out of REDCap and transform this data into FHIR resources. And I'll go in a bit more detail. I'll explain how it works and what were the goals with this tool. And finally, we used standards that are important for analytics. So one of them is the SNOMED CT expression constraint language. This is a very powerful language that lets you define value sets to constrain the search space. And I'll also talk a little bit more about this in more detail later in the talk. And we used a FHIR analytics platform, which is an open source product that we called Pathling, and it uses some standards, a standard called FHIRpath to be able to define complex analytic queries. And I'll also go into a little bit more detail later in the talk. So a REDCap form looks a bit like this, the one that's shown on the screen. Specifically in Australian Genomics, the way that REDCap was, the forms that were designed, every single flagship has forms in a single REDCap project. So this was an important consideration for the tooling that we developed. So there are a few common forms for basic information such as patient demographics and contact information for the patients, but the forms that actually capture the clinical data for each flagships, because every flagship is a bit different, so everyone developed these forms independently, but are all part of the same REDCap project. This is a user interface for our terminology server, so it lets you explore some of the terminologies that are loaded in it. So here you can see we have SNOMED CT. So it's really interesting because it lets you navigate through the hierarchy and really get a really interesting hierarchical view or more of a graph view of these ontologies. So one of the main plugins that we built for REDCap in order to overcome some of the limitations of the forms that had been developed when we joined Australian Genomics is this plugin that lets you build autocomplete style forms in REDCap by using OntoServer in the back end. So this is really important because, as you can see from this slide, whenever you have free text, that is really, really hard to process and to do analytics with. So we've done some work in some of the flagships to transform these previously, so these fields that were previously captured as free text into coded data using this plugin and using SNOMED CT and other terminologies as well. So a little bit of more detail around Redmatch. So I guess our main goal with this tool was to be able to have a way to transform REDCap forms into FHIR resources without having to use a programming language. So that's kind of the aim of this tool. So someone that obviously knows a little bit about FHIR, but is not necessarily someone with a programming background, can write some rules in a domain-specific language and you know, be able to extract things built in REDCap into a common FHIR model. So this is kind of the typical structure of a rule. You have a condition that tells you if the rule is executed or not, and then if that evaluates to true, then you create a FHIR resource and you populate attributes mostly based on values that come from the REDCap form, but also could be just static literals that are needed in some cases. So this is an example of what you get once you use this tool. So you start with a REDCap form, like the one you can see on the left. This actually was the first case study we did with this tool for the somatic cancer flagship in Australian Genomics. And once you define all these rules and put them through the platform, then you end up getting a really nice and standardized FHIR model, as you can see on the right-hand side. So in this case, you can see that we have 3 encounters. Each encounter has a series of observations and conditions, and then everything is tied back to the patient. And the last bit of technology is the FHIR analytics server. So as I mentioned before, what this component does is extends the normal searching capabilities of a FHIR server with additional capabilities. And I will explain this a little bit in more detail later in the talk, but this just really provides more robust searching capabilities that enables you to do analytics. So now that we have a bit of an idea and an overview of the technology and what Australian Genomics is, I will start explaining a little bit more in detail what we did with the pilot. So I'm going to start with the cardiovascular genetics disorders pilot. As Ian mentioned, this pilot is the biggest of the two. It was almost a year-long bit of work. So the cardiovascular flagship was recently expanded, and it is now expected to recruit up to 600 families. And all the patients are going to undergo whole genome sequencing. And one of the goals of the flagships is to be able to capture high-quality clinical data upfront as a prerequisite for ordering the genomic testing. So the main goal of this pilot was to determine if SNOMED CT provides adequate coverage to do deep phenotyping for cardiovascular patients. So the scope of this work was first to work with the flagship clinicians and kind of refine what they meant by the patient phenotype and define explicitly what was going to be captured in the REDCap forms. As part of this process, we had to identify what terminology content was needed for the flagship. So, for example, if you define a field in the form and you know that this field is meant to capture some very specific type of information, for example, cancer type, then you would need to define a value set for that form. So that is a significant amount of the work that we did in terms of identifying what was the terminology content that was required. Also, as part of this work, we needed to assess if there were any content gaps, and if they were, then of course triage these content requests, and also act as a liaison point with our local release center. So this is one of the an extract, because it's a really long form, an extract of the form we developed for the cardiac flagship. And as I mentioned before, one of the interesting bits of work was defining all the value sets that go into each of the fields that are using the autocomplete style plugin. So this is an example of one of those fields. So you can see that in this case we have an autocomplete style field to capture the detail of congenital heart disease when the user clicks on the yes button and says, yes, this patient does have indeed a congenital heart disease. So we can capture more detail, but then of course this autocomplete field needs to be bound to an appropriate value set. So some of the work we did was to actually defining those value sets. And one of the most important tools in that definition of the value sets was the SNOMED CT expression constraint language. So in many cases, it is enough to define value sets simply by saying this value set corresponds to all the children of a particular concept. But there are also some circumstances where that is really not enough. And what the expression constraint language does is that it enables you to use all the description logics that have been used to define SNOMED CT. So you can actually refer to all the attributes and everything inside SNOMED to come up with complex definitions of value sets. So this was actually very useful while developing some of these value sets for the cardiac forms. The other really important bit of work that we did with this flagship was coming up with a pattern that allows you to capture data whenever a clinician looks for a concept in SNOMED CT, but is unable to find it for any reason. So it could be that a synonym is missing, for example, or it could be that a concept is new and hasn't been modeled in SNOMED. So the pattern we came up with is what is shown in this screen. So we have an autocomplete style search box, but we do not show the field that lets you capture free text that's shown at the bottom until the clinician actually starts typing in the autocomplete style search box and doesn't find the results and explicitly indicates that no results were found. To do this, we use a functionality in REDCap that's called branching logic. So only if those not found element is selected, then this additional field is displayed. The reason for this is that we actually wanted to motivate the clinicians to actually look for the concepts. If they have the possibility of entering free text up front, then they might not bother looking for a concept. So we just wanted to promote the clinicians actually going to the effort of looking through the underlying terminology, in this case, SNOMED CT. In most cases, the concepts are there. So, the more complete that the terminology is, the more likely is that the clinicians won't be frustrated by trying to look for a concept in it. So the results from this initial pilot were drawn from a data set of 276 patients. So this project was started in April 2019, and we took a snapshot of the database in June 2020. The analysis found that the majority of the concepts that the clinicians wanted to capture were already in SNOMED CT, and therefore we concluded that it's a suitable choice for this clinical area. We did find a few gaps, but all of these were highlighted in a report that is available from SNOMED International, and we submitted all of our findings to SNOMED International. So that is the first pilot. The second pilot is quite different. So this is a smaller scale pilot, as Ian mentioned, and we did this pilot with the KidGen flagship, which is interested in working with inherited kidney diseases and has the aim of improving diagnosis, management, and treatment of these diseases. So in this case, the pilot had two main goals. One was to improve the quality of the data that had already been collected. So it's a bit different from the first pilot, because in this pilot, the forms had already been built and had already been populated with data. So we had to improve those forms instead of help building them from the ground up. And the second goal was to explore analytics use cases using SNOMED CT and FHIR. So we divided this project in two phases. The initial phase was mostly aimed at improving the quality of the data in the forms. So what we did was to use the plugin that I showed before and replace some of the free text fields with coded data. So this was done by one of our clinical terminologists, and the difficult cases were consulted with the flagship leads. So in the end, we ended up with all of the data set fully coded for the diagnosis field, which was really the relevant field. And I'll explain a little bit in more detail what we did. And in the second phase, we explored some of the analytics use cases that were relevant for the flagship leads. And I'll go into a little bit more detail on exactly what we did. And we implemented this use case using the CSR analytics platform. So this is actually similar to what I showed earlier. So we have, in this case, 3 fields that were of interest. There was actually 3 diagnoses, and I'll explain a little bit more in depth what the flagship leads were interested about this diagnosis. But these are diagnoses that happen at different points in time to the patient's journey, and they contain free text. So what we did was we added an additional section in the form. So we didn't obviously replace the original data. We just added this additional coded fields at the end of the form. And as you can see from this slide, we're using our plugin to pretty much recapture that initial information, but using coded data using SNOMED CT. Then we transformed a subset of the form into a FHIR model, and the FHIR model is pretty much what you can see here on the screen. It's very simple. We have a patient, we have 3 encounters, and each encounter has a condition that is actually used to represent the diagnosis that was done at each encounter. The issues we found in the initial phase of the project were mostly around modeling. So we found some concepts were in the wrong hierarchy, some concepts had missing relationships, and there were a few missing concepts and synonyms. And again, in this case, we produced a report and we submitted this to SNOMED International. The second phase is where we used this FHIR model to do analytics. So the main idea that the flagship leads had was to be able to visualize how the diagnosis of a patient going through a multidisciplinary clinic evolved through time. So when a patient was part of this project, it was first referred to a multidisciplinary clinic. So at referral, they were diagnosed. Then the patient was assessed at the clinic. A second diagnosis was produced using all the information available at that stage, but without any results from genomic testing. And a final diagnosis was done again by this multidisciplinary team once the genomic testing results were ready. So the idea was to visualize how these diagnoses evolved throughout this patient's journey. This is important because it helps calculate the diagnostic yield, and it also helps evaluate the model, which is something that was being tried out. So it was important to have some solid data around how well this model works. So our analytics platform is actually trying to overcome some of the problems that are prevalent in healthcare, and most of these have to do with not having a standardized model. So often when some analytics are required, people come up with bespoke, complex, and difficult models, and then multiple models are built, sometimes even within the same organization. So the main idea around our platform is, instead of actually using a bespoke model, just rely on FHIR, which is a standardized model. So some of the questions that really we were trying to answer with this platform was, how do we simplify the task of querying health data? The second one that's really important is, how do we expose clinical terminology to analytics users in a way that's easy for them and they don't have to become experts in the field? And again, how do we lower the costs of the implementation? And this is, if you have a bespoke model or multiple bespoke models, it's really hard to get data to look to conform to all of these models. If we have a single one, then I think it's much easier. So yeah, the solution is really what we implemented in our platform. And again, as I was mentioning before, it's based on FHIR. So this means that you already know the model. This is a good model for people that have domain knowledge, and it also lowers the cost of integrating data that might be already available in FHIR. So the reason, one of the reasons to target FHIR and SNOMED CT for analytics is mostly that FHIR provides first-class support for SNOMED, and FHIR already provides a framework for defining a domain model that is machine-readable, extendable, and reusable. And it actually includes some of the core elements needed specifically for Australia and also for other countries. So this is an overview of the functionality of the analytics server. I won't go into a lot of detail given the time constraints, but I think the best way of looking at it is mostly as a pivot table as an API would be a way to describe it for those of you who know Excel. One of the key standards that is used in the implementation of PathLink, which is the name of our analytics platform, is FHIRpath. So this is just a graph traversal language that describes arbitrary expressions related to FHIR data structures. So FHIR is a complex model that has a graph-like structure, so it's not a flat model. So this language just lets you, refer to different bits of the model in a standardized way. So it's critical for the implementation of our platform. This is an example of FHIRpath. I'm going to go very quickly through this because we don't have a lot of time left. So this is an example of how you would get a list of qualification codes for all practitioners involved in an encounter. So as you can see here, it's just a complex path that traverses the graph model. And in terms of the analytics platform, the most important operation, I think, would be the aggregate operation. And again, this is mostly what provides the functionality of a pivot table as an API. So it takes a set of expressions that define aggregation functions, groupings, and filters, and returns a set of groupings and aggregate function results. Once you get these results back, you can drill down in each group result to individual records. This is an example that again, I'll go a bit quickly through it, but it lets you, so you can see the different steps in building first the filters. Then this is another filter that's saying that we're only interested in patients over the age of 65. Then you can define groupings. So in this case, we're grouping by the presenting problem category and doing a mapping to a different code system. In this case, we're grouping again by intensive care facility. And finally, we're aggregating, and we really want the median in minutes to solve this particular query. So as you can see, this is a very powerful query language. And when you execute this query, what you get, the results you get are something like this. So you'll have, in the columns, you'll have the groupings that you defined. So in this, only the data that it matches the filters is included. And then the final column will be the median function that is calculated based on the results. So we use this functionality to implement this and produce this really nice Sankey diagram for the flagship. So what this is really showing you is, in a very compact and concise way, is showing you how these diagnoses evolved for all the patients. So as you can see, on the left, we have the diagnoses that were done at referral. Then you can see that a small percentage of those diagnoses actually became more specific when they got to the multidisciplinary clinic. So that in itself shows that the model does provide value even without genomic testing. And then when you look at the right-hand side, you'll see that once the genomic testing results are received, then of course lots of things happen. You get some diagnoses become more specific and get confirmed. Some others still remain provisional as expected, not because genomic testing is not 100% accurate. And then you get some of them are refuted, and you also get some. There's also, this diagram is also showing cases where the results are still pending, so they haven't been received for the patients. But you know, the interesting thing is that you'll get a very concise view of this for this particular use case. Thank you. So yeah, that's it for now. So happy to take any questions.
Well, thank you very much, Alejandro, and thank you, Ian, for a great presentation. We've got some interesting questions, and let me start. I'm going to take it from the top, both of you. So there's a question from Kristen that asks, how did you choose which elements from the FHIR resources you need for your model? And could you use them in other disease areas?
Yeah, I think I'll go ahead and answer that one. So yeah, that is a very good question. So I guess part of the process in Australian Genomics also involved building what it's called the FHIR implementation guide, and that is precisely the process that decides which elements of the generic FHIR model are relevant for this particular area and for the use cases that are interesting for the clinicians and the flagships. So we've taken more of a bottom-up approach, I guess. So we've started with what the flagship leads have told us is relevant for them, and we're building this implementation guide based on that. But as you say in your question, I think we do want to make it as generic as possible in terms of being broadly usable in the area of genomics. And yeah, so you really need to build this implementation guides in FHIR for them to be, useful, because otherwise, plain FHIR does let you, it's very unconstrained. So you have many different ways of representing things, and also there's lots of information and lots of resources in FHIR that are, very important for certain use cases, but not necessarily for if you have, like, for example, if you're working in genomics, so not everything is relevant. So this helps you obviously simplify the model. If you just look at all of FHIR, it is very complex. So it is a process of just really looking into your use cases and determining which elements are important and documenting this.
That's great. Thank you very much. Ian, I'm going to assume you don't want to add to that answer.
You are correct.
Okay. There's a kind of linked question, I guess, which is on the more technical side. So it's probably again for you, Alejandro, which is, does your model allow you to capture data in the different hierarchies in SNOMED CT? And can you give examples of which hierarchies you've been taking data from?
Yeah, so the FHIR model has typically certain fields that are coded, and those coded fields are the ones that you can actually populate using concepts from a terminology. So, for example, SNOMED CT. Part of the process of building the FHIR implementation guide is precisely defining what you want to, what sort of concepts you want to put in those fields. But typically, like base FHIR doesn't limit what you can put into those fields very much, except in very rare cases. Most of the time, it gives examples, or it lets you extend the bindings of what the concepts that you want to put into those fields. So yeah, so to answer the question specifically, yes, you are allowed to capture data not only from different hierarchies, but in theory you could add even codes from different code systems. I'm not sure if that would be a good idea, but you have the possibility of doing that. And one of the really interesting things of using SNOMED for this is that you can define these value sets using the expression constraint language. So it's really powerful in the sense that you can really build very complex value sets with this language. But yes, yeah, you can mix and match hierarchies. You just have to define exactly what you want in that field, and that's part of the process of building the implementation guide.
So, and there's a sort of linked question, I think, that's just come up at the top, which which is essentially saying that if you simplify queries too much, then you may lose some of the granularity and important information for describing a disorder. Have you got views how you might manage that by maybe using expression constraint language or putting in specificity?
I don't think I follow that question, to be honest. I'm assuming this is referring to querying the analytics platform, and I wouldn't say that the queries are, you're not really simplifying the queries. You're just standardizing the model that is used to answer those queries, but the queries can be quite complex. And in fact, as part of the work we've done with the analytics platform, we've implemented some very complex examples. The thing is, even if the queries are not simple, if you're using a standardized model, then it's easier to reuse these queries later. So it's not. I don't think it's about simplifying the queries. Most of the queries, if you're after something that is complex in nature, then you still have to write a complex query. But I think that the fact that you can reuse the queries later if you have the same model is where you have you can gain lots of, well, you simplify the process because you don't have to rewrite a query, for example, based on a different model. So that saves you time and money. I hope that was what the question was about.
Yeah, no, I'm sure you've made a very good stab at it, Alejandro. We've got to give Ian some work to do. So I think we all have some grasp of what you've been doing in Australia. But the question for Ian is, how do we get some spread internationally?
So I think the approach we're taking, or the driver for doing the pilots in the first place, was to really showcase the utility of SNOMED to meet the genomics use case. So we're working obviously to publicize it. So if some of you are on Twitter, you'll have noticed Twitter feeds going around highlighting the report that came out of the pilots. But we also work alongside GA4GH, which is the Global Alliance for Genomics in Health. So we're showcasing the pilots and the reports from the pilots through GA4GH. So GA4GH has just over 500 organizations now as members. So getting the word through GA4GH feeds through into communities that we wouldn't normally talk to as a SNOMED community. So you're really looking at getting that information out to genomics researchers and getting them to understand the utility of SNOMED, how it can be used, particularly to allow them to access data directly from EHRs to support a lot of the discovery process within the genomics research. Also, we are a partner organization with ISO. So ISO have recently set up a subcommittee specifically looking at genomics. So we feed into that, and these reports will also go through there. But it's also important to say that these pilots it's not the start and the end. So we will continue to look at some of the utility around the way SNOMED can be used in the genomics space. One area particularly which would leverage a lot of the analytics functionality within SNOMED is to start to work with biobanks and look at biobanks storing data in SNOMED format, but also using the structure of SNOMED and the ontological principles to actually start querying really quite scalable and sizable databases. There are certainly a couple of large databases within Europe that I'm aware of, because part of my role is customer relations for Europe, and we'll be looking at working with those as we move into 2021. But it's something we need to build on. There is no easy answer. And because you're looking at two very distinct communities, the clinical community and the genomics community, there are some challenges around communication, definitely. I hope that helps.
I agree. I'm going to ask a question that's not on the list, I'm afraid, and I'm afraid it's aimed for Alejandro, and I'm not sure, Alejandro, if whether this is in your toolbox, but I think one of the challenges for current clinicians is exploring what happens to carriers of monogenic diseases, and whether symptoms that they develop over a lifetime may be related to their carrier status. I come from a generation where basically we believed that most carriers were symptom-free, but I think as we explore more the issues of penetrance, the more we need to know about how that varies in different populations and in different individuals. Do you think we can use the phenotypic information in SNOMED to explore that question?
Yeah, I think that's definitely possible. Of course, the challenge there is to capture that information, right, throughout the lifetime of the individuals. But you know, I think that the tools are there, and yeah, I think the part of the goals of this pilot, one of the goals was to try to figure out if SNOMED could be used to do this sort of what it's called, to refer to sometimes as deep phenotyping, which is capturing the phenotype to the level of detail which can enable this sort of analysis. So I think SNOMED can be used, but of course, actually capturing that information, I think it's what's the challenge.
Yeah. So as always, it's back down to the nurses and doctors to record the data. So one last question, which is really about, do you think health professionals need to understand anything about FHIR, or is it just something that's in the background that they don't have to worry about?
Yeah, that is a good question, and it's a difficult question. I think it would be good for health professionals to at least have an idea of what FHIR is and what advantages it has. But I think, to be honest, it should sit in the background for most health professionals, and it all depends. Some health professionals are like to know a little bit more about this, or they might be more inclined to know about the technology. So I think, yeah, it would be definitely useful. But ideally, any good technology just sits in the background and does a good job and enables the professionals to do their work properly. So yeah, no, it is a difficult question. I'm not sure what Ian thinks about this.
I think from my perspective, I think clinicians need to understand the utilities of SNOMED. The utilities of FHIR. You don't need to understand the detail and be able to write FHIR queries, but you do need to understand the utility, to understand the benefits and how you can use it. And if you have a technical team supporting you, in some ways, what questions are you going to ask to be resolved? So understanding what can be done with FHIR, I think, is important. Understanding it to a technical level, definitely not, I would have thought.
That's great. We've used up all our time. So Alejandro, thank you very much for a great presentation. Ian, thank you for being also a great presenter and answering the questions very precisely. I'm sure that the audience will have enjoyed that. And may be able to pick up some of the threads by using the network channels and network rooms. So thank you all very much, and see you all later. Bye-bye, everyone.
Bye. Thanks very much.